So, good evening to all the participants, resource person in this Sunday and in the Science Dialogue series. I know you are very dedicated. A speaker is also dedicated, and attendees are also dedicated so that they are attending on Sunday. So, I welcome you all in the Science Dialogue series. At least you are willing to learn. Out of thousands of research scholar, student, you are ahead than those who are not attending. So as you all know that this is free webinar series. Purpose is to increase knowledge pool of the society, especially in biosciences stream or the subject which helps the bioscience. And as you all know, today's topic, this is like interdisciplinary topic related with chemistry, but its application is in biological science also. As you all know, most of you know that we give e-certificate also provided you attend complete lecture and fill the feedback form we will share in this chat box here. So don't ask me feedback link after this lecture and don't leave the lecture within that. Otherwise, you will not see the that feedback link. I request you all to attend and interact with the speaker. That is main purpose here. Here you will get more opportunity to interact because in a conference seminar only a speaker get time for 20 minutes to 30 minutes. Within that time they have to deliver and interact. Here you have enough opportunity to interact. In between also you can interrupt the speaker. Interrupt means you can unmute and ask question if you find that something is difficult or something is necessary to ask. So this is about Science Dialogue series. More than 110 free lecture has been organized under this Science Dialogue series. As you all know, today's speaker is very dynamic and we know each other, but sir has approached me to deliver lecture. That is courage. That courage is required because sometimes I approach, but I expect that researchers should be proactive to deliver lecture, those who are confident on his or her topic. I approach several people, but sometimes I only get consent from 10%, 20% people because they don't have a ready topic. They have spent 10 years, 20 years in their research or academic field, but they don't have a topic ready to deliver even for 1 hour to open lecture. Classroom teaching is different, online teaching is different. So it needs more courage to deliver to unknown people. So I welcome Dr. Arun Sharma sir for accepting formal invitation from my side to deliver lecture to our participants. Our participant means no one is my student and all of the participants are my student. Means all are Indian citizens and sometimes outside India also. Some of the participants may be from subcontinents. So with this, I am happy to introduce today's speaker. Uh, Dr. Arun Sharma sir is Associate Professor at Kota, Career Point University. And before joining, he has also done his postdoc from BITS Pilani, Goa campus, and PhD from Jivaji University, Gwalior. And during PhD, most of his work he carried out at DRDO. As you all know that this is Defense Research Organization. So he got opportunity to work there as a SRF Senior Research Fellow. He did his BSc MSc from Ajmer because I think he's native of Ajmer. Anna sir?
Yes, sir. Yes, sir.
Yes. So welcome, and I heartily appreciate your time, your effort, and your you can say proactive approach. With this brief introduction, sir is also a member of several you can say scientific society, uh, including this Royal Society. He has published in Royal Society of Chemistry. So he is basically from chemistry background, but he's working on biological things. He's applying that chemistry principle on biological things like a biosensor. So, sir, welcome in the Science Dialogue series. Now, platform is yours.
Thank you so much, sir. Professor Dr. Gopal, sir. I am very much thankful to you for giving me such a wonderful platform, and I am really very happy to deliver my talk with such a knowledgeable and qualified audience who are willing to join my lecture, and I'm very much happy to give an informative talk on next generation biosensors and the technology, because the technology is very important nowadays, and many countries are improving their technology. And in life science, we are not we we are not can say that we have no any kind of resources. We have everything in our country, and we have a center of excellence, we have establishments. I also from a state university graduate and postgraduate, and I got the opportunity. I qualified some competitive exams, and I also encourage my students to set a goal in their life, and target their dream, so that they can achieve the target. And such platforms will provide you the opportunity to learn from the experts, and I am feeling very much grateful, and I again thankful to Professor Dr. Gopalji to giving me such a wonderful opportunity. Without wasting your time, I am starting my lecture on the today's cutting edge technology, which is on biosensing. Now I am sharing my presentation. So I think that the presentation is visible to the audience.
यस यस विजिबल, उसको हाइड कर दीजिए, वो यहाँ जो नीचे दिख रहा है ना, जस्ट हाइड दैट लाइन.
Right. Okay. Okay. Right.
Yes,
Right.
yes, yes.
Yes. Yes.
Thank you. It's
So
visible.
now sir, I shall continue my presentation.
Yes, yes, yes. Please continue.
Thank you. So the title of the presentation is next generation electrochemical biosensing, which you already know. Okay, this slide basically introduces the core theme of the presentation, which is the integration of nanotechnology with electrochemical biosensing for the next generation diagnostic tools. Basically, it sets the stage for discussing how nanoscale materials and devices are transforming conventional biosensors, making them more sensitive, selective, and suitable for smart diagnostics. In the coming slide, this is the topic which I've selected for my talk. Uh, this slide outlines the main segments of the presentation. It provides a roadmap that includes the introduction, basic introduction of the field, the scientific approaches and techniques used, applications of nanotechnology in academics and research, and my personal contributions, my own contributions in the area of my interest, and potential future and advancements, which is very necessary and help the audience to understand the structure and flow of the session. Now come to the nanotechnology. Nanotechnology is a terminology which is combined with two basic terms, nano and technology. Nano means very small, and you can say that it is 1 billionth of a meter. And technology, you all know that about the tools, devices, methods for practical purposes, which establish the scale at which nanotechnology app operates and presents the audience to understand the significance of manipulating materials at such a small scale, tiny scale. So you can see these the nanomaterials and the nano dimensions are not visible by the naked eye, you use we need some specific equipments, apparatuses to explore the nano shape and size of the nanomaterials. Yes, it is very common, and we know about all these things. These are the basic things, and you know that the nanotechnology is in the range 1 to 100 nanometer. If it is beyond 100 nanometer, it is not nano, it is micro. Okay, if it is less than 1 nanometer, then it will comes to the angstrom level or the pico level. Okay, so basically this slide reiterates the formal scientific definition and reinforce how working at the this net scale allows for novel applications in electronics, materials, energy, and biology. It further sets up the transition to biosensing applications because we are using the application of nanotechnology. Nanotechnology, a lot of applications. One of the application which I am working in my lab is sensors or immunosensors. Okay, so you see that the nanotechnology is now in spintronics devices, in the photoluminescent applications, smart optoelectronic properties, right? So, but we are focusing of particular to the biosensing technology because biosensors are in very much demand after the COVID-19 outbreak, and you see that in previous pandemic situation also like anthrax outbreak and the influenza H1N1 outbreak, botulinum neurotoxin, staphylococcal enterotoxin, which is one kind of food poisoning, which uh, basically I have worked on staphylococcal aureus bacteria. It is a foodborne pathogen, which I will explain in my coming slides. First, I will introduce what is biosensor. Okay, so biosensor, in this slide, you can see the mechanism of the working of biosensors. Okay. So biosensor basically it has two components. One is the bioreceptor or the biorecognition element, which is we can use as a bioreceptor as a like antibody or in the enzymes, and transducer. Transducer may be any kind of device which is a like piezoelectric or the electrochemical any kind of electrode we can use. In this case, we are using electrochemical method, electrochemical technique. It established how biosensors play a pivotal role in medical diagnostics, environmental monitoring, and food safety. Okay, so biosensor, one of the in biomolecule like antibody. We know about antibodies. You know, it is immunoglobulin, IgG, right? So this antibody is basically a Y shape. It is a generally as per the protein structure. It is a Y shape, and there are some bonds there between the small small arm and the big arm. Okay, and there are some detection sites which detects the foreign part, analytes having the antigens, because in the samples it detects the analytes like in our body fluid. If some foreign particle enters in our body, the antibody activates and it target the antigens, and the rate of rate constant, or you can see, because I am the chemistry person, so I basically come to the kinetics, and it is a very fast reaction. The antibodies capture the antigens very fastly and makes and form the immunocomplexes. Immunocomplexes means the combination of the antibodies and with the antigens, so it will form the immunocomplex, and these are very stable. Complexes are very stable. Okay, so now come to the types of biosensors, because you see that people will confuse that how there are variety of biosensors available in the market. So in what kind of principle they are working basically. So according to the principle, we name the biosensors, we categorize the biosensors in colorimetric. So in the colorimetric means the color change. You are measuring the change in color. So such a biosensor like the you can see the pregnancy strips. Okay, if the color is changing at a particular time when you put the sample on the paper, then it is a colorimetric biosensor. It gives you the confirmation test. Okay, and now come to the electrochemical biosensor. Electrochemical means it gives you some electrochemical signal, amperometric signal or the voltammetric signal, right? So you can detect the signal with the help of biosensor, and you can also find out the how much the you can quantify the analytes which is present in the sample by using this kind of biosensors. Optical biosensor and the same term, the optical signals they will monitor. Piezoelectric biosensors means they are mass sensitive biosensors. And like the quartz crystal microbalance, you can understand about QCM technology, and the SPR technology is there also. Piezoelectric means piezo means pressure, and some pyroelectricity means thermal changes when it produces the electricity. And piezo means when you apply some sample, and if it is loaded with the analytes, it will change some pressure, and you monitor that pressure change, and the change in pressure will generate the electricity, and you can get the signal. And there are similar lot of products available in the market for the detection of or the identification of the infectious disease, like glucose biosensor, glucometer for the diabetes patients, right? So that kind of equipment, basic equipment, handheld devices, portable devices, miniaturization. We are basically, nanotechnology means the we are miniaturizing the simplifying the process. We are reducing the cost, and time is also saving the time also, because time taking techniques are not fit for the market. Customers and the patients and the like medical, in the medical sector, hospitals, doctors, they basically need small devices, so they can easily carry it anywhere in field, also on site, off site. They can use it very easily, simple to use. Okay. So this kind of technology we are working. And now my field, I come to the slowly I am coming, I am putting the attraction, the audience to my area of interest, electrochemistry. I'm electrochemist. Okay, so the electrochemical biosensors is the area of my, because I have worked on basically fluorescent biosensors and the electrochemical biosensors. So first we know about the electrochemistry. Electrochemistry means electrons. You know, everywhere electrons are there around the nucleus. Electrons are present in the orbitals. Okay, and they are unpaired electrons, pairs, some paired electrons, and the movement of electrons will generate the electricity. When the electrons shift from one end to the another end, so the electrode which is giving the electrons will oxidize, and that this is the donor of the electrons. And the similarly in parallel there is another electrode which receives the electrons, and that particular electrode will reduce, and that is called anode. Okay, so there will be cathode and anode, and when this circuit will complete, it will form the electrochemical cell. And basically the purpose of electrochemical biosensor, which is we can say it is a device that detects the target analytes by measuring electrochemical changes. Okay. And this is the advantage of the electrochemical techniques, like the cyclic voltammetry, linear C voltammetry, DPV. You can see here, they are cost effective and having the potential of miniaturization. It is the central type of biosensor, which is uh, I will discuss in the rest of my presentation. Now come to the basic techniques of the electrochemistry: cyclic voltammetry, linear C voltammetry. Okay, these are the electrochemical techniques. In cyclic voltammetry, in brief, it is called CV, not curriculum vitae. It is cyclic voltammetry. If I am a teacher, students may confuse with the abbreviations. We are using the abbreviations. We just to explore the things. LSV means linear CV voltammetry. SWV means square wave voltammetry. These are the electrochemical techniques, and we use these techniques by the help of potential state, galvano state. Okay, it is a uh, fundamental equipment which is used for the biosensing applications, right? So it is available in my lab, and you can also use, and whenever if you want to use that kind of instrument in your electrochemical analysis, you can reach to the Gopal sir also and me also directly, so we can help you to um, analysis of your electrochemical analysis of the samples which is provided by you. Okay, so this is also one of the angle you can explore in your future research or in your current research also. So these are the techniques. Another techniques also, there are some fundamentals behind it. What is the pulse width and the pulse time, period time, because time is very important. And you can just when you are analyzing any kind of sample, and it if it is taking more time, then you can get the chance, there will be chances to get the contamination, and you will get the false signals also, and doctors will may not get the right therapy to the patient. So it is very important to get the results within the nanoseconds, microseconds, or in few seconds. You will, if you will get the results, then it will be helpful for the patient to save his life, and it is also helpful for the doctors and the medical personals to take care of the patient, that particular patient, and give us the right treatment at the right time. So these are all about the techniques. Now come to the application of the biosensors. There are so many applications in the forensic, in the medical diagnostics, in the food analysis, in the drug development, crime detection, medical diagnostics, environmental monitoring, quality control, industrial process control, and detection of biological warfare agents. I am doing all these things. I did food analysis also. I did biological warfare agents in DRDO, and in the BITS Pilani Goa campus, I worked on cancer biomarkers. So I will also explore the work which which I'm presently doing and published in the journals which are mentioned in my introduction. So from that also you can understand that what kind of work is going on, and what are the things we can start in our laboratory, and we can collaborate also. We can uh, this is the platform where we can touch to the profile, and find out that what kind of possibility will be there, like some kind of internship if you want to do from my laboratory, so that it will be beneficial for the audience, for the participants. Now come to the approaches used to design nanomaterials. There are two kinds of approaches which is generally used for the synthesis of any kind of material, nanomaterial, right? Top down approach and the bottom up approach. Top down means you have a bulk material, and you are reducing the size of the material, and it is a top down means because you have a bulk material and you are reducing the size, and come to the atomic level. And atomic level is very small, because the atoms are come into the angstromic scale, pico levels. In between, the powder, like the micrometer size powders, are like very small particle size, and they have large surface area. When you are reducing the size, it will you will you will get the nanoparticles. In opposite, there will be the bottom up approach. Bottom up means from the ions, you are you are colliding, there will be agglomeration by adding some reagents, and you will get the stabilized nanostructures.
Visakha Singh, mute your mic. Visakha Singh, mute your mic.
Yes, sir. Okay, sorry.
म्यूट करो विशाखा. हाँ, प्लीज कंटिन्यू सर.
Right, right.
आपका भी थोड़ा एयर का आवाज आ रहा है क्या कुछ वहां पर, या वाइड वगैरह कहीं कुछ हो रहा है? सम
Yeah,
थोड़ा सा कभी.
okay. Let me check. I
ओके, आप कंटिन्यू करिए
give me a minute.
प्लीज.
Give me a minute. I just off it. I think now the
हाँ, अब ठीक है, अब
yeah,
ठीक है, अब
right,
ठीक है.
right now. So I think there if there is any query, any discussion till my presentation, then I'm open to audience to ask something. If
हमने
they,
लिख दिया है, वैसे चैट में भी हमने लिख दिया है. इफ दे नीड एनी, है ना, सो अदर दे कैन राइट इन चैट बॉक्स और दे कैन अनम्यूट एंड
yes, yes.
स्पीक.
I'm very happy to if I get the questions from the audience, so that it will be like interactive session. So
यस.
I will be happy to give the suitable answers. Thank you. Now if sir give me permission, then I will proceed to the coming slides.
Yes, please proceed.
Okay.
I am giving all permission to all the participants to interact anytime if they
Right,
have
right.
any question, है ना?
Okay,
So
okay.
पूछ लेंगे वो.
Okay, okay.
जी जी
Now
जी
come to the stability of the nanomaterials, because the nanomaterials, nanoparticles are very small, as I told. So it is a very crucial to stabilize the nanomaterials. If the nanomaterials are not stabilized, you will get the precipitate or the coagulation, sedimentation, and in that case, you cannot use that that material for your whatever application you are going to analysis, you are going to experimentation. For such a crucial point, it is very important to add some stabilizers, like the electrostatic stabilization will be there. You can use the steric stabilization. There will be some ligands, some kind of material which you can use to stabilize your nanoparticles. There will be some cross linkers are available in the market, and you can also design as according to your application. Because I am particular looking for the biosensing application, so I have to selectively identify the stabilizer which is bio is available, bioactive, and bioaffinity with the biomolecules. Okay, so they will not deteriorate the natural condition of the biomolecules, like the antibodies, DNA, right? The bioprops, so they will not going any kind of structural changes with the biomolecules. So it is very important to select a suitable stabilizer to stabilize your nanomaterials. Now coming to the tools and techniques, there are so many analytical techniques available also in my laboratory, and there it is available. It is highly recommended to give the characterization, to get the characterize your material, because without characterization you cannot say that you have synthesized, you successfully synthesized right nanomaterial. So you have the data. If you have the data, scanning electron microscopic data, transmission electron microscopic data, atomic force microscopic data, spectroscopic data, there are so many spectroscopic techniques, x-ray diffraction techniques, zeta potential, dynamic light scattering analysis. Okay, and this kind of techniques are available. And if you are using these techniques, so you can say that you have successfully synthesized nanomaterial. They have some basic principles behind SAM, TAM, and AFM. I have shown here in my slide the difference between the all these three techniques, the microscopic techniques, SAM, TAM, and AFM. Okay, in SAM, the scattered electrons, because the name is also says the scanning electron microscope, scattering electron microscope. Okay, where the scattered electrons are analyzed. Okay, it is, but it is having limitations, like resolution is very low, and you need sample preparations. But in case of AFM, no sample preparation is required. You can directly put the sample in the AFM. Okay, but there will be the chances to damage of the probe, the photo detector. Okay, so the cantilever, because it is a very at angstrom level tip, so it will be the possibility to damage of that kind of tip. So that kind of limitation is associated with any kind of microscopic techniques. So these are some basics. Now come to the spectroscopic techniques. Spectroscopic techniques available in also in my laboratory. I have FTIR techniques, UV visible techniques are there, Raman spectroscopy is there. Okay, photoluminescent spectroscopy you can also do. So these kind of techniques are available in my laboratory, and these are the basics of the UV visible spectroscopy. And it depends on the size of the material. When the size increases, you will get the red shift. Okay, to the higher wavelength. Redshift means you are getting the peaks at the higher wavelength range. And but the if the size of the material is small, you will get the blue shift. Blue shift means the you will get the peak intense peak at the smaller wavelength region. Okay, so this is the spectroscopic techniques which is generally used for the determination of the size of the material, and also right to check about its fluorescent properties and the optical properties, right? Like IR will give you the nature of the bond, and the what kind of linkages will be there in your nanomaterial. Because if you are using any kind of technique for the stabilizing your nanomaterial, it is stabilized or not, the confirmation will be given you by the spectroscopic techniques. Now come to the DLS techniques. Raman spectroscopy. You all know about all these things. Okay. Raman is very simple technique, and it is discovered by, or the investigated by Dr. C.V. Raman. He is a well-known scientist. Now come to the XRD. XRD is very common technique for the sample analysis, for the determination of the size of the material. It is also there, like Sasher equation is there. It is not about the biological people, but if they are aware, they should aware about all these techniques. What is the principle behind it? What is zeta potential? Okay, there will be the charge. If the charge difference is more than minus 30 millivolt to plus 30 millivolt, you can see there will be the electrostatic repulsion between the materials, and due to the electrostatic repulsion, the material will get stabilized in their particular condition. So a biochemist must know about all these basic things. Now come to the application of the nanotechnology in academics and research. Right. So these are some photographs which I captured in from the websites. These are not my own photographs. So no need to worry about. But these are they are realistic images. You can, if you are working in any kind of lab. Okay, if you get some postdoc opportunity in state of the art laboratories, you will definitely get this kind of infrastructure, because infrastructure is very important. You have the setup of all kind of experimentation tools you have to available for your material science studies, and for your biochemical studies, or in biochemistry, also in biosensing apps. There are some courses provided by in nanomaterial science by IITs and the premier institutions, center of excellence in India also, and out of like in Coursera, in MOOCs courses, NPTEL. There you can select these courses available on the websites in different agencies. State of the art research projects you can submit for the funding purposes to CSIR, to ICMR, DBT also, DST also. So you can get funding if you don't have. We have some funding in our projects, and we are working on the some in some projects in my own laboratory. My scholars are working. I have scholars from Nigeria, from Zimbabwe, from South Africa, and uh, other countries also. I have some students in Netherlands. Sorry, I miss from Netherlands. I have some abroad scholars doing PhD under my supervision. So there is also opportunity to get doctoral degree from any university where you want to continue your doctoral studies. Okay, so you should be very careful to select any kind of institutions before starting your PhD. You must aware about the facilities available in that in the laboratory, the expertise of your supervisor. Okay, because it is very important. If your supervisor is very helping in nature, your PhD will work on will be going on very smoothly. If and in the opposite case, you know that what kind of situation will be there in case of the PhD scholars. So now come to the next slide about my focus to come to the my basic contribution in the particular field. This is data is already published, so I have no worried about the data. I'm already showing the data which I already published in the journals, and I will also show you my publications here. So these particles prepared by scholars, not by me. These are my scholars work. They are already get published their work. Okay, so you can see the beautiful materials, the structures, gold, platinum, bimetallic nanoparticles, zinc sulfide, nanoflowers. You can see the flower like structure of the zinc sulfide, nano rod like structures. Some images you can see the nanoporous zinc oxide, platinum nanoparticles. Okay, so you can see that these kind of materials we are synthesizing in my laboratory, and scholars are working. So it shows the enthusiasm and the curiosity of the students to synthesize novel materials, smart materials, and application of these materials for the biological warfare agents to to decontaminate the bioweapons, because the COVID-19 is also like we can say it is used as a bioweapon, and still world is suffering from the various strains of the COVID-19. And recently we are also in news and media, the new strain of the COVID-19 is spreading. So this is problematic, and we have to be very careful and take security measurements. Okay, and we know that you already know about all these things, the regular washing your hands, wearing of masks, keep distance, don't go to the populated area. So this kind of things will be there. And you can see the categorization of the biological warfare agents according to their toxicity and their LOD and their problem, how much they can, and they have potential to kill the people, right? So this kind of images are there, like the viruses are very small, but their impact is very big, and it will come to the death of the people, many people. Okay, so that is why they are called bioweapons. I worked, my scholars are working on staphylococcal enterotoxin B. It is a foodborne pathogen. And the LD50 lethal dose and the ethical dose, lethal dose, ED50 value is mentioned here. It is a foodborne pathogen, and it is very resistant to the temperature fluctuations. It is very stable virus or bacteria, you can say. And it has two basic routes of exposure, like from the inhalation route or the ingestion route. You can one can expose with this bacteria. Now it disease in humans, this bacteria causes so many diseases in humans. It via in inhalation route, there will be a high fever, chills, muscle pain, everything is mentioned here. And the images you can also see that how much impact it can produce on human body. So we are working on the human models, and so it is a very good exposure for the students, that they will get such kind of experimentation. So they are very much happy, and feeling grateful to work in such a uh, excellent area, right? So now there are some methods for the detection of the SEB, and these are the methods which are already reported in the literature, because it is very important to give the importance of the literature. We are, if the scholars are not going through proper literature survey, they will get confused in their studies when they start their experimentation. So I think that I and recommend my scholars, and to every scholar who is willing to pursue his PhD or any kind of new area of exploring new something, then they should go through the literature survey thoroughly, and use internet also. Now there are some AI tools are also available. So scholars are recommended to use AI tools, and use the AI tools in your research, not for the writing of your research paper. You write research paper by your own thinking, and your accurate data, your original data you should publish. You should not uh, you should not find the data through AI. I do not recommend for the data analysis by using AI tools, but I recommend researchers to use AI tools for literature survey, and for the current requirement and the demand of of your work in the for the society, right? Now the objectives of the study which my scholars have been they are doing, and they are the rights provided by my scholars, which I am happy to share with you people, so that you can also know that these are the basic objectives which in my lab students are working on, basically electrodes. And we are using conventional techniques for the comparison purpose, like enamel immunodorment assay, ELISA, and the fluorescent techniques also using. And they are synthesized some nanoparticles and the quantum dots. Quantum dots are also three dimensional, zero dimensional nanomaterial, and having the quantum confinement of the antibodies is so this is the basically which has been published in Pack Factor 3.8. They are showing here in electroanalysis, and this is the paper they have published for the detection of this antigen SEB. Okay, and they have synthesized that material which I already shown in my in the previous slides. And this is the mechanism when you how to put the antibodies. There will be physical adsorption, there will be rabbit anti-SEB antibodies, then they wash the electrodes, then there will be the blocking with the 3% bovine serum albumin, then washing, continuous washing, and the incubation, okay, for a particular time. Then after they put the antigens and may form the immunocomplex, then there will be a sandwich ELISA, and after that they have put some alkaline phosphatase linked antibodies, right? There is a for the I think it is a for the enzymatic enzymatic degradation for the alkaline phosphate LP. ALP is the substrate for this kind of this this enzyme. And when you put the electrode which is coated with the nanoparticles, you will get the ultra sensitive and the fast response. And because there will be electron transfer when the substrate gives the product ascorbic acid. Ascorbic acid is vitamin C, and this alkaline phosphatase catalyzes this reaction, and it quantitatively determines the amount of the antigen present in this sandwich immunocomplex. And you will get the signals, electrochemical signals, and particularly there will be a table here, zero point 0.1 nanogram limit of SEB they have detected. So it is a very smart and the quantitative analysis by using the electrochemical techniques. If any kind of if any question is there regarding this paper, so
कुछ
I am happy
क्वेश्चंस
to
हैं इसमें लिखे हैं एक ने जेबी आर दिस डाउन साइड्स ऑफ यूजिंग नैनो मटेरियल टू ट्रीट डिजीज वो कुछ हेलो
hello.
हाँ एक क्वेश्चन इसमें
Yes, please
आर
please.
देयर डाउन साइड्स ऑफ यूजिंग नैनो मटेरियल टू ट्रीट डिजीज ऐसा क्वेश्चंस हैं बाकी तो इंट्रैक्शन वाले हैं वन क्वेश्चन इज व्हाट आर of the precipitates formed from poor characterization of nanomaterial or individuals. आप
Mm-hmm.
कंप्लीट कर लीजिए देन यू कैन रिप्लाई व्हाट
Right. I think that I have to complete first. My video is not there.
हाँ वीडियो नहीं चल रहा आपका इंटरनेट स्लो है
Internet issue
एंड
is there.
यस पीपीटी दिखा दीजिए
I think that I have come to the PPTs.
यस 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 कंप्लीट के बाद उसको डिस्कशन
Mm-hmm. Now, this is my our next work, which is published in RSC Advances, and this work is also gives us great, wonderful results for the detection of that bacteria SEB at the zero point zero 0.01 nanogram level. So it means you can say that 10, it is up to 10 picogram per ml. We detected this particular bacteria by help of the quantum dots. And this is the electrochemical immunoassay. In case of flyzi also, there is a 30 picogram per ml, means electrochemical techniques is more sensitive as compared to the fluorescent techniques. बुला रहे हैं तुमको एक बार जाना. So this is the basically work which is already published in the RSC Advances. Now come to the another work which is published in Biotechnology Reports, and in this work we have not received high sensitivity, but we detect the bacteria till up to 1 nanogram per ml. And but here we change the material only, by the rest of the procedure is the same. Okay, so there is only the change of the material, and the same procedure is applied, and we get the we get the detection limit up to 1 nanogram per ml. This work is also new and recently published in New Journal of Chemistry. It is one of the journal of Royal Society of Chemistry, and here we have used graphene, uh, graphene and the chitosan and the gold nanoparticles for the coating electrodes. And after the coating of the electrodes, we have put the formation of the immunocomplex using the sandwich ELISA process. And for the signal amplification, we have used the ferrocene carboxylic acid labeled antibodies, and here you get the excellent signals up to 5 nanogram per ml SAB we have detected with this technique. And we have used here multi-watt carbon nanotubes and the graphene as for the comparison, and we get good results in case of graphene as compared to the multi-watt carbon nanotubes. This work is also published, has been already been published in Clinica Chimica Acta, and here we have synthesized the polymeric nanospheres for the electrochemical detection of the PA antigen, which is prostate antigen. Okay, and the detection limit we have received here 0.1 nanogram per ml for the detection of this particular antigen. Okay, and these are the nanospheres we have used for the detection of this kind of BW agent. And PSA means polystyrene acrylic acid. It is they are doped by the cadmium. Cadmium ion is is basically reduced at zero point 0.4 volts, and have provided us very good results. These are the same image of the nanomaterials. Here they are the up to 50 nanogram to 100 nanogram size of the nanospheres, which is revealed by this kind of this same image. Now the possibilities come to the possibilities for the future. Nanotechnology may make it, you can read it very easily. I just conclude my topic. The future possibilities and this presentation envisioning the future of nanotechnology, programmable materials, fuel efficient devices, nanorobotics, and smart diagnostics. This presentation basically stress the transformative role of nanotechnology, which will play across industries and human health. So whatever we are doing, it should be beneficial for the society, beneficial for the country and the world. So because in the life science sector, it is our duty to design novel materials and look their application for futuristic generations. Thank you so much. Now I have completed from my side. If I am very much happy to give you the answers